water is basically causing salt or sugar to move down the concentration gradient. If too much water builds up inside of a membrane, osmotic pressure results. Do you remember what I said happens when there's too much osmotic pressure inside of a cell? It causes the cell to lyse or burst, which kills the cell. This is why it's really important that you don't have too much salt or sugar in your diet. It's also why when you eat sugary or salty foods, you get thirsty. Two things can happen. It can either draw water out of your cells, and then you can either drink too much water too quickly, which will cause the cells to burst, or you can simply have too much salt and too much sugar built up inside of your cells over time, which is drawing more and more water into your cells, which can create that osmotic pressure. So everything in moderation. Of course, sugar and salt aren't necessarily bad for you. In fact, you need some sugar and some salt in your diet in order to be healthy. There can be some serious consequences if you're not getting enough salt. But you don't want too much either. It causes an imbalance of the water level inside of your cell with the water level outside of your cell. So if osmotic pressure is too high, the cell can burst or lyse. So let's take a look at osmosis in action.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So your next quick question, how does osmosis relate to cells? Well, osmosis is how water gets into and out of cells. All right, let's talk about that. Structure and function of the cell membrane. So we know the structure of the cell membrane already. We've talked about it. It's a phospholipid bilayer embedded with protein pumps. So we talked about phospho, meaning phosphorus that you get from your dark green leafy vegetables, and lipids are fats, which you can get from healthy fats such as olive oil, avocados, peanut butter. So we have our phospholipid bilayer membrane, two layers, bi of course means two, embedded with these protein pumps. The cell wants to maintain equilibrium. Remember we talked about homeostasis. It wants to maintain a constant internal environment. So in order to do that, it is selectively regulating the passage of substances in and out of the cell. And it does this through a few different methods. Small molecules such as water, carbon dioxide, and oxygen pass easily through via diffusion because they're small, so they can easily pass through this phospholipid bilayer membrane. This is really good because water is a nutrient, carbon dioxide is a waste product, so it needs to get rid of it quickly, and oxygen is also a nutrient. Large molecules such as starches from the foods that you eat require use of these embedded protein pumps for facilitated diffusion. Which is a type of passive transport. So what this means is that it doesn't require additional energy. It's just too large to fit through the membrane via osmosis, so it needs to use the protein pumps to move it through. But it's not using energy besides kinetic energy to move through those protein pumps. Let's take a look at facilitated diffusion in action.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
We have two types of diffusion that we've discussed now. We have simple diffusion, which is small molecules just simply diffusing through the phospholipid bilayer. If it's water, then it's diffusing through the process of osmosis. It happens to small and non-polar particles. What polar means, so if you remember the water molecule, so we know that water kind of looks like Mickey Mouse, right? We have one hydrogen molecule bonded to two oxygen molecules, and these oxygen molecules are negative and the hydrogen is positive. It's considered polar because the charge is uneven. There's more negative charge than there is positive charge. That's what makes this molecule polar. So simple diffusion happens with small and non-polar particles. Facilitated diffusion happens through a transport protein that's embedded in the phospholipid bilayer membrane, and it can happen with large or polar particles. All right, so let's take a moment and go back and talk about active transport. Active transport being the transport that requires additional energy. There are two types of active transport. The first type involves protein molecules in the cell membrane that act as carriers aiding in transportation. We just talked about those protein molecules being involved in passive transport, but they can also be involved in active transport. The second type of active transport is endocytosis. So what we're seeing here, this is our phospholipid bilayer membrane here. We have these embedded proteins. We have different molecules on the outside. And this is one of the smaller molecules that is just simply diffusing through the membrane without a protein pump. These molecules are either larger or polar, and they need facilitated diffusion, so they need the help of these protein pumps to move through the membrane. Active transport is again using that same protein pump, but extra energy is needed, and it could be because these molecules are moving from an area of low concentration to an area of high concentration. And when that happens, additional energy is added, and it's often in the form of ATP. We've talked a little bit about ATP. If you really want to know about ATP, take AP Bio. But ATP is the body's energy currency. It's what's created when our body breaks the food that we eat down into molecules that the body can use as energy. Okay, let's talk about endocytosis. There are two types of endocytosis. The first is phagocytosis, which literally means cell eating, and it's when white blood cells are engulfing foreign pathogens. So germs, bacteria that will make us sick, the white blood cells come along and quite literally eat them. So if my hand with a germ, this would be the white blood cell that comes along and engulfs it. And it does that by the white blood cell's membrane folding around the large molecule or the germ so that they can be taken in by the white blood cell, which results in a vacuole. We talked about the vacuole organelle in the cell that will hold the molecule until the lysosome breaks it down and gets rid of it. The other type of endocytosis is called pinocytosis, which means cell drinking. So there can be fluids that contain large molecules, such as DNA or proteins, that are taken into the cell through the same methods. So the cell membrane folds around the liquid containing the large molecule and brings it into the cell. What we're seeing up here in this image, this is an example of endocytosis. So this would be a white blood cell and it's engulfing a germ. The germ's saying, oh no, because it's about to die, which is good for you because you won't be sick. Okay, that's diffusion and osmosis and a little bit more detail about the cell membrane. Make sure that you really understand this because we're going to be doing a lab on diffusion.